Greetings, tutor. Excited to dive into one of the most fundamental and frankly crucial topics for our biology exam today, cell cycle and cell division. It just feels like this chapter always has a lot of intricate details, and the specific stages that can get a bit confusing, especially when you're prepping for NEET. Could you perhaps help us understand it thoroughly?
Namaste. Absolutely. That's an excellent choice for us to tackle. The cell cycle and cell division chapter is indeed a cornerstone of biology. It's not just for your class 11th and 12th board exams, but it's also a really high-yield topic for NEET and, various state-level medical entrance exams too. Understanding how a single cell can give rise to a complex organism with millions of cells is fascinating, right? And equally important is knowing the precise sequence of events. So today, we'll take a comprehensive deep dive. We'll focus on clarity, exam relevance, and making sure you can really visualize each step, even without diagrams in front of you. So let's unpack this. Okay, let's begin with the very basics. What is the cell cycle? Think of it simply as the life story of a cell, right from its birth, its formation, all the way to its own division into two new daughter cells. This entire sequence, this journey, that's the cell cycle. Now, the core concept here, and it's fundamental, is that all organisms, even the largest ones you can imagine, start from just a single cell. Growth and reproduction, these are fundamental characteristics of all living organisms. We know this. And cells reproduce simply by dividing into two. Then these new cells, these daughter cells, they can grow and divide again.
So that's how we get from one cell to, us, with millions of cells.
Exactly. This growth and division happening over and over allows that single cell to form structures consisting of literally millions of cells. It's the basis of multicellularity. So if we want a precise definition, maybe for board exams, the cell cycle is the sequence of events by which a cell duplicates its genome, synthesizes other cell constituents like proteins and organelles, and eventually divides into two daughter cells. That definition covers all the key aspects.
That makes sense. Duplicating the genome, making more stuff, and then dividing. So it's not just about splitting, but a whole orchestrated series of steps, like you said. But then what are these stages or phases of this cell cycle that you mentioned? How is it broken down?
Excellent question. To understand this coordinated process better, the cell cycle is broadly divided into two main basic phases. Let's take this step by step, because getting these two main phases clear is the first hurdle, right? The entire cell cycle. We divide it into two primary phases. First, there's interphase. Now, this is the phase between two successive M phases. It's often called the resting phase, but honestly, that's very misleading. It's far from resting. It's actually the period of intense preparation. This is where the cell gets ready for division. It undergoes significant cell growth and, crucially, DNA replication, all in a very orderly manner.
Okay, so interphase is the long prep time. What's the other phase then?
The second main phase is the M phase. This stands for mitosis phase. This is the phase where the actual cell division or mitosis occurs. It's the more dramatic part you might see under a microscope. Let's consider a typical example, just to get a feel for the timing. Human cells in culture. They divide roughly once every 24 hours. Now here's a surprising fact that often trips students up, so pay attention to this for competitive exams. In that 24-hour cycle for a human cell, the actual cell division, the M phase, lasts for only about 1 hour, just 1 hour out of 24.
Yeah, only an hour.
Yes, which means interphase occupies more than 95% of the total duration of the cell cycle. Almost all the time is spent in preparation.
95%, that's huge.
It is, but remember, this duration isn't fixed across the board. It varies. For instance, yeast. It can complete its whole cell cycle in just about 90 minutes. Much faster.
Right, so the 95% is a general idea for human cells. Okay, so if interphase is so long, what exactly happens during this long preparatory phase? Are there subphases within interphase itself?
Precisely. Interphase, being so long and absolutely critical, is further divided into three distinct subphases. Let's break these down carefully, as understanding what happens in each is fundamental. Okay, interphase is subdivided into three phases. First is the G1 phase. That stands for gap 1, or you can call it the first growth phase. Timing-wise, this phase is the interval between the end of the previous mitosis and the beginning of DNA replication, so right after the cell is formed. Activity-wise, during G1, the cell is metabolically very active. It's doing its normal job, growing continuously in size. Most organelle duplication also occurs here, making sure the new cells have enough mitochondria, ribosomes, etc.
But no DNA copying yet.
Crucially, no. The cell does not replicate its DNA in the G1 phase. The DNA is still in its original state. Now, let's talk chromosome and DNA content, because this is a classic exam point. If a diploid cell starts with, say, 2N chromosomes and 2C amount of DNA, in G1, it stays 2N and 2C. Nothing has changed there yet.
Okay, 2N, 2C, and G1. Got it. What's next?
Next comes the S phase. S stands for synthesis. The key event here is obvious from the name. This phase marks the period when DNA synthesis or replication takes place. This is the only time DNA is synthesized. So, what happens to the DNA content? During S phase, the amount of DNA per cell doubles. If the initial amount in G1 was 2C, it increases to 4C.
Right, doubles the DNA, so it becomes 4C. But what about the chromosome number? Does that double too?
Excellent question, and here's the crucial distinction. There is no increase in the chromosome number during S phase. If a cell had a diploid number, 2 ends of chromosomes at G1, it will still have 2 N chromosomes after the S phase.
Wait, how? If the DNA doubled?
Because each chromosome now consists of 2 identical sister chromatids joined together at the centromere. Think of it like photocopying a book, but keeping the original and the copy bound together. It's still counted as one book or one chromosome until that binding, the centromere, splits later on.
Okay. Like 2 identical twins holding hands counts as 1 unit for now. That helps. That's a common confusion point for NEET, right?
Absolutely. DNA content, C, doubles, chromosome number N stays the same in S phase. Remember that. Also, just a specific point for animal cells. During S phase, DNA replication happens in the nucleus, obviously, but the centriole also duplicates out in the cytoplasm. That's important for spindle formation later.
Okay, central duplication too. Good point. So G1 growth, S phase DNA synthesis. What's the last part of interphase?
The last part is the G2 phase, or gap 2, the second growth phase. This phase follows the S phase and comes just before the M phase kicks off. During G2, the cell continues to grow a bit more, but the main activity here is synthesizing proteins specifically needed for mitosis. Things like the proteins that make up the spindle fibers. It's the final preparation push.
Final checks and building the machinery for division.
Exactly. And in terms of content, the cell still has 2 N chromosomes. Remember, the centromeres haven't split, but the DNA content is now 4C because it was duplicated back in S phase.
So G1 is 2N, 2C, S phase goes to 2N, 4C, and G2 stays at 2N, 4C.
Perfect. You've got the sequence of changes in N and C through interphase.
That book analogy for the 2N, 4C thing in S phase was really helpful, but this brings up another question. What about cells that don't divide? Like, you hear about nerve cells or heart cells not dividing in adults. Where do they fit into this cycle? Do they just get stuck somewhere?
That's an excellent follow-up, and you've hit on 2 very important concepts for exams. First, yes, the book analogy or your linked twins idea, perfect for understanding why 2N chromosomes can have 4C DNA. They are duplicated but joined. Now, your question about non-dividing cells, that's exactly where the G Varroa phase comes in. It's called the quiescence stage. Some cells in adult animals, like heart cells or many nerve cells, they don't seem to divide at all after they mature. Other cells, maybe like liver cells, might divide only occasionally, maybe to replace damaged cells or if there's a need.
So they leave the cycle.
Precisely. These cells exit the G1 phase. They don't proceed to S phase. Instead, they enter an inactive stage called the quiescent stage, or G0. Now, inactive here means inactive with respect to division. They are still very much metabolically active. Heart cells are pumping, nerve cells are firing signals. They're doing their jobs.
Okay, so G0 isn't dead, it's just not preparing to divide anymore.
Exactly. They no longer proliferate, meaning they don't divide unless, and this is key, unless they are called upon to do so. Depending on the specific requirement of the organism, maybe due to injury, some G0 cells can be signaled to re-enter the G1 phase and start dividing again.
That makes sense for tissue repair.
It's crucial for understanding tissue repair and maintenance, yes. And one more point, relevant for NEET especially, about where mitosis happens. In animals, it's typically only in diploid somatic cells, but there are exceptions. Male honeybees, which are haploid, actually undergo mitosis to make sperm.
Haploid mitosis, interesting.
Yes. Whereas plants, they're more flexible. Plants show mitotic divisions in both haploid and diploid cells. Think about the alternation of generations you studied, both the gametophyte, haploid, and sporophyte, diploid stages. In many, plants can grow by mitosis. That's a classic comparative point.
Right, plants do things differently. Okay, I think I have a good grasp of interphase and G0 now. All that preparation leads up to the main event, the M phase. What happens there?
Okay, great. Now let's move to the M phase. This is really the most dramatic period of the cell cycle. It's where the actual division happens, and it involves a major reorganization of pretty much all components of the cell. Mitosis is also known as equational division. Remember that term? Why equational? Because the number of chromosomes in the parent cell and the daughter cells remains the same. If the parent is twoukant, the daughters are twoukant, equal.
Okay, equational division, unlike meiosis, which we'll get to.
Exactly. Now, although for convenience, we divide mitosis into 4 stages of nuclear division called karyokinesis, it's really important to understand it's a continuous, progressive process. There aren't sharp stops between stages. They flow into one another. We just put labels on them to help us study the key events.
Right, like chapters in a story, not completely separate books.
A good way to put it. So the M phase involves 2 main parts happening in sequence. First, karyokinesis, that's the division of the nucleus, leading to the separation of the daughter chromosomes. It's all about the genetic material. Second, cytokinesis. This is the division of the cytoplasm, which follows karyokinesis and divides the entire cell into 2 daughter cells.
Nucleus first, then cytoplasm. Makes sense. So what are the stages of karyokinesis?
Karyokinesis has 4 stages: prophase, metaphase, anaphase, and telophase. Let's explore each one, focusing on the key events you need to identify for exams. Let's start with prophase. This is the first stage of karyokinesis. It follows the S and G2 phases. Remember, in S and G2, the DNA was replicated, but the new DNA molecules are still long, thin, and kind of intertwined, like chromatin.
Right, like tangled threads.
Exactly. So the first key event in prophase is the initiation of condensation of this chromosomal material. Think of those long, spaghetti-like strands starting to coil up, fold, and become tightly packed. This condensation process makes the chromosomal material visible and distinct under a light microscope. They start looking like actual chromosomes. And importantly, as they condense, the previously intertwined DNA molecules become untangled. This is essential for separating them later. By the end of this process, each chromosome is clearly seen to be composed of 2 sister chromatids attached together at the centromere. That classic X shape becomes visible.
So condense and untangle. What else happens in prophase?
The second key event involves setting up the division machinery, the centrosome. Remember, it duplicated back in S phase in animal cells. Now, these 2 centrosomes begin to move towards opposite poles of the cell. As they move, each centrosome radiates out microtubules, forming structures called asters. The 2 asters, plus the spindle fibers, which are more microtubules forming between the poles, create the entire mitotic apparatus. This is the machinery that will pull the chromosomes apart.
Okay, so building the spindle. And you mentioned things disappear too.
Yes, by the end of prophase, if you look under a microscope, organelles like the Golgi complexes, the endoplasmic reticulum, the nucleolus, they seem to disappear. They actually break down or disperse. And critically, the nuclear envelope also starts to disintegrate. This breakdown is vital because it lets the chromosomes out into the cytoplasm where the spindle fibers can attach to them.
Wow. So prophase is about condensing chromosomes, building the spindle, and breaking down the nuclear boundary. Quite a busy stage. What happens next after the nuclear envelope disappears?
Excellent transition. The complete disintegration of the nuclear envelope marks the start of the next stage, metaphase. And metaphase is often the easiest stage to spot in images because of its very characteristic feature. Since the nuclear envelope is gone, the condensed chromosomes are now spread throughout the cytoplasm. By this stage, chromosome condensation is complete. They are at their thickest and most visible. This is why metaphase is the stage where the morphology, the shape and size of chromosomes is most easily studied, useful for karyotyping.
Okay, maximum condensation. What's the structure like?
Each metaphase chromosome is clearly made of 2 sister chromatids held together by the centromere. You can see the X shape very clearly. Now, a really important structure for NEET. On the surface of the centromeres are small, disc-shaped structures called kinetochores.
Genetitors. Okay, what do they do?
They are absolutely vital. They serve as the specific attachment sites for the spindle fibers. The microtubules of the spindle grab onto the kinetochores.
The connection points?
Exactly. And this leads to the defining characteristic of metaphase: alignment at the equator. The chromosomes are moved by the spindle fibers right into the very center of the cell. They all line up along an imaginary plane called the metaphase plate or equatorial plate.
All lined up in the middle.
Perfectly aligned. And the attachment is specific. One chromatid's kinetochore is attached to spindle fibers from one pole, and its sister chromatid's kinetochore is attached to fibers from the opposite pole. This ensures they'll be pulled apart correctly.
That makes sense for equal division.
So, key features of metaphase: spindle fibers attached to kinetochores, and chromosomes align at the metaphase plate, ready for separation.
Okay, metaphase is alignment at the center. So if they're all lined up, the next logical step has to be their separation, right? How does that happen? Does it happen all at once?
You're thinking exactly like a biologist. The separation is indeed the next dramatic step, known as anaphase. And yes, it's quick but powerful. The key event, right at the start of anaphase, is the simultaneous splitting of the centromere of each chromosome, Remember that single point holding the sister chromatids together? It splits instantly. This is the moment those linked twins finally let go.
The splits, so they become individual chromosomes now.
Precisely. Once the centromere splits, the two sister chromatids are now referred to as daughter chromosomes. And they immediately begin their rapid migration towards the two opposite poles of the cell. They move away from the equatorial plate. The centromere leads the way, directed towards the pole, while the arms of the chromosome trail behind. This often gives them that V or J shape as they move.
Okay, centromeres split, chromatids separate and move to the poles. What about the chromosome count here? You mentioned something tricky earlier.
Right, the exam point. During anaphase, temporarily, the chromosome number in the entire cell doubles. If the cell was 2N before anaphase, it briefly becomes 4N chromosomes within that single cell boundary, because each separated chromatid is now counted as an individual chromosome moving to a pole. The DNA content, however, remains 4C overall until the cell divides.
Temporary doubling of chromosome number within the cell during separation. Got it.
It's a crucial detail for numerical questions. So, key events of anaphase. Centromeres split, sister chromatids separate, and these daughter chromosomes move to opposite poles.
That splitting and moving sounds very precise. So once they reach the poles, is that the end of nuclear division? What happens to the chromosomes then? Do they just stay there?
Once the separated daughter chromosomes arrive at their respective poles, we enter the final stage of karyokinesis, telophase. And telophase is essentially the reverse of prophase. It's about putting everything back together to form two new nuclei.
Reverse of prophase, okay.
Yes. First, the chromosomes that have reached the poles begin to decondense. They uncoil, lose their distinct shapes, and become less visible, reverting to that diffuse chromatin state again. Then, a new nuclear envelope develops around each cluster of chromosomes at the two poles. This forms two distinct daughter nuclei.
So the nucleus reforms. What about the other stuff that disappeared?
Good recall. The nucleolus, Golgi complex, and ER, which disappeared in prophase, begin to reform within each new daughter nucleus. Everything gets rebuilt. So, key events of telophase. Chromosomes cluster at poles and decondense. A nuclear envelope reforms around each cluster, forming two daughter nuclei. And the nucleolus, Golgi, and ER reappear. Karyokinesis is complete.
Okay, two new nuclei formed. But the cell itself is still one big unit, right?
Exactly. Karyokinesis is done, but cell division isn't finished until the cytoplasm divides too. That process is cytokinesis. Right, cytokinesis is the final step, ensuring the cell division is fully completed by separating the cytoplasm. Mitosis segregates the chromosomes, karyokinesis, but cytokinesis divides the cell itself into two daughter cells. Now, how this happens differs between animal and plant cells. That's important.
Okay, how does it work in animal cells?
In animal cells, cytokinesis happens by the appearance of a furrow in the plasma membrane. Imagine pinching a balloon in the middle. This furrow gradually deepens from the outside inwards and eventually joins in the center, pinching off the cytoplasm into two. This is called a cleavage furrow.
Pinching in from the outside. Okay. And plants, they have that wall, right?
Exactly. Plant cells have a rigid cell wall, so they can't pinch. They use a different mechanism. In plant cells, wall formation starts in the center of the cell and grows outward towards the existing side walls.
Inside out. How?
It begins with the formation of a precursor called the cell plate. This forms in the middle. This cell plate grows outwards and eventually fuses with the side walls, forming a new wall that separates the two daughter cells. This cell plate represents the middle lamella, the layer between the two new cells.
Center outwards, forming a cell plate. Got it. What about organelles like mitochondria?
Good question. During cytokinesis, organelles like mitochondria and, in plants, chloroplasts get distributed roughly equally between the two daughter cells. They don't divide precisely, but they segregate so both cells get a share. Now, there's an interesting exception to cytokinesis always following karyokinesis. Sometimes nuclear division happens, but the cytoplasm doesn't divide immediately.
Oh, what happens then?
This leads to a multinucleate condition, a single cell with multiple nuclei. This is called a syncytium.
Syncytium. Multinucleated cell. Any examples?
The classic example often asked in exams is the liquid endosperm in a coconut. That coconut water is basically a giant syncytium.
Wow, really? Okay, that's a memorable example. So animal cells pinch, plant cells build a wall. That's clear. So wrapping up mitosis, what's its overall purpose again? Why is it so important for us?
That's the perfect question to consolidate everything about mitosis. Let's look at its vital significance. Hashtag high five. Significance of mitosis. Mitosis, this equational division, is incredibly significant. Here are its key contributions, often tested. 1. Growth of multicellular organisms. This is the big one. We start as a single zygote, How do we become complex adults? Millions and millions of mitotic divisions. It's the basis of development.
Right, growth from one cell.
2, maintenance of nucleocytoplasmic ratio. As a cell grows, its volume increases faster than its surface area, and the cytoplasm can become too large for the nucleus to control efficiently. Mitosis divides the cell, restoring a favorable ratio, ensuring efficiency.
Keeping the balance right.
3, cell repair. This is crucial. Mitosis replaces cells lost to injury or normal wear and tear. Think about your skin. The outer layer cells are constantly replaced. The lining of your gut is constantly renewed. Blood cells are constantly replenished, all thanks to mitosis.
Like fixing damage and replacing old parts.
Exactly. 4, continuous growth in plants. In plants, specific regions called meristems at the tips, apical, and sides, lateral cambium, undergo continuous mitosis throughout the plant's life, allowing them to grow much longer and thicker than most animals can.
Meristems, right.
Yeah. And finally, 5, genetic identity. Mitosis produces daughter cells that are genetically identical to the parent cell. Same chromosome number, same genes. This ensures genetic stability during growth and repair. Perfect copies.
Growth, balance, repair, plant growth, and identical copies. Okay, that covers mitosis well. Now, you mentioned the other type, meiosis, for sexual reproduction.
Yes, exactly. Now that we've thoroughly covered mitosis, let's shift to meiosis. It's a completely different type of cell division, but equally fundamental, specifically linked to sexual reproduction, its main purpose. Sexual reproduction involves the fusion of 2 gametes, sperm and egg, for example. Each gamete needs to have half the normal number of chromosomes, a haploid set. Meiosis is the specialized division that achieves this reduction. It takes a diploid cell with 2 N chromosomes and produces haploid daughter cells with N chromosomes.
Having the chromosome number, so it's reductional division.
Precisely. Because it reduces the chromosome number by half, meiosis is called reductional division. This is the key contrast with mitosis, which is equational. This is vital for the life cycle. Meiosis produces the haploid gametes N, then fertilization fuses 2 gametes, restoring the diploid number 2N in the zygote. This keeps the chromosome number constant for the species generation after generation.
Makes sense. Prevents the chromosome number doubling each time. Where does it happen?
We see meiosis during gametogenesis, the formation of gametes in plants and animals. It occurs in specialized diploid cells in the reproductive organs.
So meiosis is for making gametes, reducing chromosome number by half. A human cell with 46 chromosomes makes gametes with 23. How does this halving actually work? Does it also have phases like mitosis? Is it more complicated?
Exactly right on the numbers for humans. And yes, it has phases, but it's more complex than mitosis because it involves 2 sequential cycles of division, meiosis the first and meiosis the second. But, and this is critical, there's only 1 round of DNA replication, which happens before meiosis starts.
1 replication, 2 divisions. Okay.
Key features to remember. 2 cycles. Meiosis I and Ti, 1 DNA replication before meiosis the sex. Meiosis involves the pairing of homologous chromosomes and, very importantly, recombination or crossing over between them. This is a major source of genetic variation.
Crossing over. I've heard that.
We'll get into it. And the final product. At the end of meiosis the second, you get 4 haploid cells. So the phases are grouped. Meiosis light, Prophase I, Metaphase I, Anaphase, Telophase 1, and meiosis the second. Prophase 2, Metaphase 2, Anaphase 2, Anaphase 2, Telophase 2. Let's break down meiosis I first. This is where the chromosome number is actually reduced. And it starts with a very complex phase, Prophase 1. This is typically much longer and way more complex than mitotic prophase. It's so complex, it's subdivided into 5 phases based on how the chromosomes behave. Ready?
Okay, 5 substages for Prophase 1. Let's go.
1. Leptotene. Chromosomes start becoming visible, compacting begins. Pretty standard start. 2. Zygotene. Here's where it gets unique. Homologous chromosomes, 1 from mom, 1 from dad for each pair, start pairing up side by side. This process is called synapsis. They're held together by a protein structure called the synaptonemal complex, like a zipper. The paired structure is called a bivalent or a tetrad because it has 4 chromatids.
Synapsis. Pairing of homologous chromosomes.
Got it.
What's next?
3. Pachytene. This is often the longest stage. The tetrads are clearly visible. And crucially, this is where crossing over happens. You see structures called recombination nodules. These are the sites where homologous chromosomes exchange segments of genetic material between their non-sister chromatids.
exchanging pieces between mom's and dad's chromosomes.
Exactly. It's an enzyme-mediated process involving an enzyme called recombinase. This crossing over leads to genetic recombination, shuffling the genetic deck, creating new combinations of alleles. This is a massive source of genetic variation. It's usually completed by the end of Pecciting.
Wow, okay. Shuffling the genes, that's huge. Okay.
More. Diplotin. The synaptonemal complex dissolves, the zipper unzips. The homologous chromosomes start to separate, but they remain attached at the points where crossing over occurred. These X-shaped attachment points are called chiasmata.
Chiasmata, the crossover points.
Yes. In some cells, like developing egg cells, oocytes in some animals, diplotin can last for months or even years. A long pause. 5. Diakinesis. Final stage of Prophase's I. Chiasmata move toward the ends of the chromosomes called terminalization. Chromosomes are fully condensed, the meiotic spindle forms. The nucleolus and nuclear envelope break down, transition to metaphase I is complete.
Phew, the prophase I is intense. Leptotene, zygoting synapses, pachytene crossing over, diplotin, chiasmata visible, diakinesis, terminalization. Okay, what happens in metaphase I?
Right, metaphase I. Similar to mitosis metaphase, but with a key difference. Here, the bivalent chromosomes, the homologous pairs, align on the equatorial plate. Not individual chromosomes, but pairs.
Pairs line up in the middle.
Yes, and the spindle fibers attach such that microtubules from one pole grab one homologous chromosome of the pair, and microtubules from the opposite pole grab the other homologous chromosome. Sister chromatids face the same pole.
Okay, setting up to separate the pairs, which must happen in...
Anaphase first. This is the core reductional step. The homologous chromosomes separate and move towards opposite poles, but, and this is critical, the sister chromatids remain attached at their centromeres. The centromeres do not split in anaphase first.
Ah, so entire duplicated chromosomes move to the poles, just separated from their homologous partner.
Exactly. So each pole receives a haploid set of chromosomes, N, but each chromosome still consists of 2 chromatids, so the DNA content at each pole is still 2C. The chromosome number is halved here.
And chromosomes, but 2C DNA at each pole.
Yeah.
Okay, reduction achieved. Then telophase 1.
Telophase 1. The nuclear membrane and nucleolus might reappear. Cytokinesis usually follows, forming 2 haploid cells. This pair is called a dyad. Chromosomes might decondense slightly, but not fully, like an interphase. And between meiosis I and meiosis II, there's a brief stage called interkinesis.
Interkinesis, like a mini interface.
Sort of, but very short-lived. And the absolute key point here, no DNA replication occurs during interkinesis. The chromosomes entering meiosis II already have 2 chromatids.
No S phase between meiosis I and II. Very important. Okay, so meiosis hyokineso reduces the homologous pairs.
Yeah.
What about meiosis II? He said it's more like mitosis.
Exactly. Meiosis II is initiated almost immediately after cytokinesis I. And yes, in contrast to meiosis II is very much like a normal mitotic division, just happening in those 2 haploid cells produced by meiosis I.
Mitosis of haploid cells. Okay. Let's quickly run through the stages. Prophase 2. If the nuclear envelope reformed, it disappears again. Chromosomes recondense if they relaxed a bit. Spindle forms. Metaphase 2-1. The individual chromosomes, each still with 2 sister chromatids, align at the equator of each cell, just like mitotic metaphase. Spindle fibers from opposite poles attach to the kinetochores of the sister chromatids.
Okay, individual chromosomes line up now.
Anaphase II. Now the centromeres split. This is the key event here, just like mitotic anaphase. The sister chromatids separate and move to opposite poles. They are now individual daughter chromosomes.
Centromere split, sister chromatids separate. Yes, and again, temporarily, the chromosome number doubles within each cell as the chromatids separate. If the cell started meiosis II with N chromosomes, each 2 chromatids, 2C DNA, during anaphase II, it momentarily has 2 chromosomes moving apart, still with 2C DNA total in the cell. Telophase 2. Meiosis ends here. Nuclear envelopes reform around the chromosomes at each of the 4 poles, 2 poles in each of the 2 dividing cells. Cytokinesis follows, the result, a tetrad of cells, 4 haploid daughter cells. Each of these 4 cells has a haploid number of chromosomes and a haploid amount of DNA, C. The final state needed for gametes.
4 haploid cells, each N and C. Perfect.
Perfect.
That clarifies meiosis II. So bringing it all together, what's the huge significance of meiosis again, besides just making gametes?
Yeah.
####8. Significance of meiosis.
Great question to summarize. The significance of meiosis is profound, especially for sexually reproducing organisms. 1. Conservation of chromosome number. As we discussed, meiosis is essential for maintaining the specific chromosome number of a species across generations. By halving the number in gametes, 2nn, fertilization restores it, n plus n minus 2n. Without it, chromosome numbers would double each generation, which is unsustainable.
Keeping the species number stable.
2. Increase in genetic variability. This is perhaps even more impactful from an evolutionary perspective. Meiosis dramatically increases genetic variation in offspring.
How again?
Primarily through two mechanisms in meiosis. The first, crossing over during Prophase E packaging, which shuffles alleles between homologous chromosomes. Second, the independent assortment of homologous chromosomes during Anaphase E, which maternal or paternal chromosome of a pair goes to which pole is random for each pair, creating countless combinations.
Crossing over an independent assortment with the two big shuffles.
Exactly. And why is this variation so important? Variations are the raw material for evolution. They allow populations to adapt to changing environments. If everyone was identical, a single disease or environmental shift could be catastrophic. Variation increases the chance that some individuals will survive and reproduce, driving evolution.
So meiosis isn't just for reproduction, it's fuel for evolution too.
Absolutely. It's fundamental to life's diversity and resilience.
That makes complete sense now. Meiosis for genetic variety and maintaining chromosome numbers in reproduction. Mitosis for growth, repair, and keeping somatic cells genetically identical. Both essential, but totally different jobs and mechanisms.
Precisely. You've summarized it perfectly. We've journeyed through the entire cell cycle, distinguishing mitosis, equational, for growth repair, producing two identical diploid cells, from meiosis, reductional, for reproduction variation, producing 4 varied haploid cells. For your exams, my strong advice is revise this again. Especially focus on the events in each substage of prophase, elliptotene, zygotene, pachytene, diplotene, diakinesis, and really track the changes in chromosome number N and DNA content, C, through all stages of both mitosis and meiosis. Those N and C values are absolute hotspots for objective questions in NEET and other exams.
Right. Prophase, I details, and the NC changes. Will do.
Practice past year questions. Look specifically for questions comparing and contrasting mitosis and meiosis. Try drawing simplified diagrams yourself to visualize how chromosomes align and separate differently in metaphase I versus metaphase 2 versus mitotic metaphase, and anaphase I versus anaphase 2 versus mitotic anaphase. That really helps solidify it.
Practice questions and diagrams. Good idea.
And of course, reach out if you have any doubts lingering on this chapter, or if you need another analogy to clarify any of the steps. Keep up this focused approach, and you'll definitely master cell cycle and cell division. All the very best for your preparations.